Hi everyone, welcome to part D of this lecture series on the electron transport chain. First off, I want you to pause, cut and paste this link, and watch this YouTube video. It's the same one you've watched 2 other times before, but I want you to see it again before we continue. To me, it's really important that you visualize what the electron transport chain and the ATP synthase is doing in order to fully understand the content that we're going to go over. One term you're going to hear in biochemistry textbooks is the chemiosmotic mechanism. Chemi meaning chemical and osmotic meaning osmosis or concentration gradient. Remember, osmosis is when water moves from a high concentration to a lower concentration. It's the same idea here that chemicals in the form of hydrogen protons are going to move from a high concentration to a lower concentration. And the whole purpose of the electron transport chain was to set up this electrochemical gradient, was to put more protons, hydrogen protons, in the intermembrane space to allow this chemical gradient. And then this chemical gradient has a lot of potential energy, and that potential energy is going to be used to create ATP. This figure shows the 4 steps involved in the chemiosmotic mechanism. The first 2 are related to the ETC, or the electron transport chain. So first off, the reduced substrate, or the fuel, NADH and FADH2, donate their electrons. The second step is that the electron carriers pump hydrogen protons out as electrons flow to the last electron acceptor, which is oxygen. And we went over that in the last, in part C of this lecture series. Number 3, the third step, is that energy of the electron flow is stored as electrochemical potential. So we have potential energy. And then the 4th step is what we're going to talk about more today, which is that the ATP synthase uses the stored potential energy to synthesize ATP and uses this electrochemical gradient, this proton gradient, to generate ATP. In this slide, you can see the hydrogen protons are moving through the complexes into the intermembrane space from the matrix. Note that complex 2 is not involved. All we see is complex 1, 3, and 4. This is because complex 2 does not span the intermitochondrial membrane, so it cannot move protons from one side of the membrane to the other. The hydrogen protons move back into the matrix based on chemical osmosis, high concentration to a low concentration, through the ATP synthase molecule. Another important concept we've been discussing is something called the proton motive force. This relates to the flow of electrons from NADH and FADH2 and causes the protons to be pumped from the matrix side of the inner mitochondrial membrane to the outside of the membrane. The movement of the protons is accomplished by the complexes 1, 3, and 4. And then the protons then move back into the matrix through the ATP synthase, which is a massive quaternary protein with multiple subunits, which drives ATP synthesis. Remember, the ATP synthase, it's an enzyme, so it catalyzes a reaction. And to me, the ATP synthase is a pretty amazing molecule in that it acts like a turbine. So the hydrogen protons flow through the cylindrical rod, this rotor, and it spins and it causes the spinning. So the more hydrogen protons that flow through it, the faster it spins, and the more it catalyzes the reaction of ADP with phosphate to ATP. So it's just like a turbine where a water turbine, water flows through the turbine and it spins the turbine and it makes electricity. This is similar here, that we have hydrogen protons moving down the cylindrical rotor of the ATP synthase and moving it. And it creates this movement which allows the potential energy to catalyze the reaction of ADP with phosphate to ATP. So here's another figure showing the ATP synthase. Remember, this is a large enzyme complex and it's very complicated. Remember, enzymes catalyze reactions and they have active sites which need to be exposed for the substrates to get into. So essentially there's 2 main components to the ATP synthase. We have the FO unit and the F1 unit. This picture is actually opposite than what you saw in the previous slide, so you might need a second to get yourself acquainted with the location of things. The FO unit conducts the hydrogen protons, which allows the movement of the cylindrical rotor, and it brings the F1 unit closer. It opens up the active sites for which ADP and phosphate can then go into and catalyze the reaction to ATP. So essentially, we have hydrogen protons being conducted through the FO unit, which spins the cylindrical rotor, exposing the active sites in the F1, for which ADP and phosphate go into the active sites. The ATP synthase then catalyzes the formation to ATP. And this whole process is oxidative phosphorylation. It couples the energy from the electron transport to the synthesis of ATP. So the hydrogen-proton gradient is essential because, remember, in this reaction, you see it at the top, ADP plus an inorganic phosphate plus energy. Well, that energy is coming from the proton motive force, that the hydrogen protons are flowing through the FO unit and causing the spinning of the F1 unit to expose the active sites in this huge enzyme complex for which ADP and phosphate can bind. The last thing I want to draw your attention to on this slide is 3 protons are needed to synthesize 1 ATP molecule. So that means 3 hydrogen protons have to go through the cylindrical rod of the FO unit in order for 1 ATP to be produced. So that means we need the energy from 3 protons to catalyze this reaction. So the flow of electrons and accompanying movement of hydrogen ions, or protons, is linked to ATP synthesis via this remarkable enzyme called ATP synthase. As electrons flow down the chain of electron complexes, they also pump protons from the matrix side of the inner mitochondrial membrane into the intermembrane space. As a result, an electrochemical gradient builds up in the intermembrane space. As we just saw, the process of chemiosmosis dictates that these ions will want to flow from a region of higher to lower concentration, which means that they want to flow back into the mitochondrial matrix. They do this by moving through the ATP synthase enzyme, which couples the flow of the hydrogen atoms to the synthesis of ATP. So we have a bit of a conundrum here. We have a lot of ATP that is being synthesized by the ATP synthase, but we need to move it to the cytoplasm because that's where ATP hydrolysis typically happens. Then we have a high concentration of ADP and inorganic phosphate in the cytoplasm, but we need to move that into the mitochondrial matrix to use as a substrate to generate ATP. So there are actually clever mechanisms to accomplish both of these tasks. There's something called an antiport that moves ATP out and ADP into the mitochondria, and a symport that moves both inorganic phosphate along with hydrogen ions that we know are moving down their concentration gradients. So now let's talk about the amount of protons that need to go through the ATP synthase in order to produce ATP. So overall, it takes 3 protons to make 1 ATP. But as I just said, we need to move that ATP into the cytoplasm. So that requires 1 proton equivalent to move ATP out and ADP into the mitochondrion. So there is a net of 4 protons required to get 1 ATP into the cytoplasm. So with that in mind, that we require 3 protons to yield 1 mitochondrial ATP, or we need 4 protons to generate 1 cytoplasmic ATP. We need to now calculate how much ATP is produced from 1 NADH molecule. So remember that NADH transfers electrons to complex 1. In total, 10 protons are moved out of the matrix with 1 NADH molecule, 4 through complex 1, 4 through complex 3, and 2 through complex 4. none through complex 2 because it is not a transmembrane protein. So since 4 protons are needed for 1 ATP molecule, we have 2.5 ATP are generated per NADH. So you have 10 protons divided by 4 protons needed for 1 ATP means 2.5 ATP are generated for 1 NADH. Now let's discuss the ATP production from 1 FADH2. Remember that FADH2 transfers electrons to complex 2. In total, 6 protons are moved out of the matrix, 4 through complex 3 and 2 through complex 4, none through complex 2. So since 4 protons are needed for 1 cytoplasmic ATP, we have 1.5 ATP are generated per 1 FADH2 molecule. Well, how did we get that? Well, there's 6 protons are moved for 1 FADH2. 6 divided by 4 protons needed for 1 ATP equals 1.5 ATP per 1 FADH2 molecule.